blood vessel growth key to healthy fat tissue. New research led by York University's Faculty of Health shows that inhibiting a protein within blood vessels stimulates new blood vessel growth, resulting in healthier fat tissue, adipose, and lower blood sugar levels. The findings provide key insight into how improving blood vessel growth could help to mitigate serious health problems that arise with obesity, such as diabetes. Tara Haas, a professor in the School of Kinesiology and Health Science at York University, and a team including first author Martina Rudnicki, a postdoctoral fellow in the Haas lab, investigated a process of the vascular system called angiogenesis. Angiogenesis is the formation of tiny blood vessels called capillaries. It helps to maintain normal healthy functions in tissue, particularly when that tissue enlarges. However, when the tissue expands as it stores excess fat, angiogenesis is repressed, and new capillaries fail to grow. This results in unhealthy adipose tissue, which increases the risk of developing diabetes and cardiovascular diseases. It is not clear why exactly new capillaries fail to form in obesity, says Haas. Previous research led by Haas found that in obesity, the levels of FOXO1 increase in endothelial cells, which are the main cells that comprise capillaries. What we know is that a protein called FOXO1 is present in the cells that line the inside of blood vessels, and that it can stop the development of new capillaries. FOXO1 controls how cells spend their energy, and it can force them to go into a resting state. During obesity, the levels of FOXO1 increase in capillary endothelial cells. The team inhibited the production of FOXO1 protein and monitored the behavior of capillary endothelial cells and the resultant influences of these changes on the expansion of adipose tissue. The study found that lowering FOXO1 levels promoted angiogenesis and caused a healthier adipose tissue, in particular, with a high-fat diet. Lower levels of endothelial cell FOXO1 also led to an increase in the use of glucose and less weight gain, despite consuming a diet high in fat. While we anticipated that improving angiogenesis in adipose tissue would make the adipose healthier, we were surprised that altering the levels of a particular protein, FOXO, in endothelial cells exerted such broad influence on whole-body glucose levels and weight gain. This led us to propose the novel concept that the activity of endothelial cells can alter whole-body energy balance. Haas said the research showed that removing FOXO1 made endothelial cells hyperactive, and that this enabled the formation of a lot of new blood vessels in the fat tissue. At the same time, these blood vessels consumed a lot of energy, and this in fact could alleviate common health problems that accompany obesity, such as high levels of blood glucose and circulating fats, which are risk factors for diabetes and cardiovascular disease. More research is needed, Haas says, to investigate the optimal way to target these cellular processes to obtain positive health outcomes.